So learning unit 6 on lipids is probably going to be the shortest learning unit in terms of content that's required for the in-class portion, but a significant portion of your take-home is going to have questions derived from this. So again, using sort of this chart that we made at the very end of our first learning unit, when we think about some important aspects of lipids, this chapter really is called lipids because it's a catch-all for anything that's really hydrophobic or nonpolar. We tend not to be talking about things that are true polymers where we're kind of putting monomer units together. But when we talk about triglycerides or phospholipids or fatty acids, right, we take fatty acids, we hook them on a glycerol backbone, and if we have 2 fatty acids that we hook together, we get phospholipids. If we have 3 fatty acids that we hook together, we get triglycerides. So those are kind of an important thing to note. We will talk about a few vitamins and hormones that are nonpolar, so they fall into this lipid category. But some important numbers to kind of know with regards to this chapter. These are worth about 9 calories per gram, so this is what makes it nature's favorite storage fuel. If you want to store energy, you're going to store it in the most efficient manner possible. So you're going to store it in something that puts the most number of calories in the least amount of space. Some other numbers that I'll have on a study guide for you guys have to do with some numbers related to sort of lipid panels. So what's kind of a standard triglyceride level, HDL level, LDL level, just some numbers that I'd like you to have sort of in your brain, and then we'll expand upon those when we have our take-home. So, we're going to kind of contextualize this the same way we talked about protein structure and then carbohydrate structure, is thinking about how we have linkages together to generate these biological molecules. So these aren't truly polymers, but we are going to be talking about linkages. And so the monomers, if you will, are going to be fatty acids. So we spent a good bit of time kind of dissecting what it means to be a fatty acid. Again, fatty acids have a carboxylic acid functional group on its hydrophilic head, and then some kind of a hydrophobic tail. And so we talked about all the different flavors we could have of hydrophobic tails. If they are completely sp3 hybridized carbons, it's going to be saturated fatty acids. And then we began to talk about the different ways that we can unsaturate fatty acids. So again, when we think about having double bonds in these units of unsaturation, some important parameters come into play. The first is that when we think about the stereochemistry, these are going to be of the cis configuration for almost all naturally occurring fatty acids. This is important because it adds a bend or a kink in the structure. And in a few slides, we'll see the implications for that. These double bonds are never in conjugation, and that's very different when we see these as compared to something like beta carotene or some other molecules that are colored that have units of unsaturation that are in conjugation. We tend not to see these units of unsaturation before carbon 9, so complete saturation, kind of as we get close to the hydrophilic head group. And then our fatty acids, that is those that we tend to see in our body and what we consume dietarily, tend to have an even number of carbon atoms. So all of the ones that we have kind of shown here as exemplars are 18 carbon atoms. So we're going to spend a little bit of time here and then a deeper dive in some potential take-home questions talking about omega fats. So omega, again, meaning is the last letter in the Greek alphabet. So omega really refers to these fatty acids that have units of unsaturation that begin either 3 or 6 carbons from the end. So omega-3 and omega-6 fatty acids are both essential. Again, essential meaning that we cannot make them. We need to consume them in our diet. Some information that I do want you to know about these: you don't need to really know any of the names of these fatty acids at all, but I do want you to know that omega-3 fatty acids are anti-inflammatory. Omega-6 fatty acids are pro-inflammatory. So again, omega-6s are necessary to reduce inflammation. Omega-6s are necessary to cause inflammation. And we'll expand on this a little bit when we talk about why you need omega-6s and why it's important to be able to cause inflammation. So again, thinking about what these look like on a structural level, if we have these saturated fatty acids, like we saw on this previous page here, saturated fatty acids here. What happens, these chemistries that we link them together, we'll see on a future slide here the actual linkage chemistry. But we're going to have a glycerol backbone. So we'll look at what the structure of glycerol is. And when we have three of these that get hooked up together, this is how we generate triacyl glycerols or triglycerides. We're going to abbreviate those TAGs. So we spent time on this slide kind of talking about how structures, again, afford functions with lipids here. And when we think about the potential intermolecular forces that can happen with something that's completely straight, and we think about the only kind of option for intermolecular forces, which are going to be London dispersion forces with something that's a hydrocarbon. So London dispersion forces have everything to do with surface areas that can interact. And so when we have these linear pieces, we're going to have increased intermolecular forces, which means we're going to have a higher melting temperature than its unsaturated counterparts, and so high that these melting points tend to be above room temperature, and so these substances tend to be fats at room temperature. The rest of these molecules down here represent either fatty acids or, again, triglycerides that are made from fatty acids that have 1, 2, or 3 units of unsaturation. Each one of these units of unsaturation introduces a bend in the molecule such that we've reduced the amount of surface area that can interact by these London dispersion forces. So these triglycerides tend to make molecules with decreased intermolecular forces, lower melting points, and they tend to be liquids at room temperature. So again, we'd refer to these as oils. So we had a nice infographic that kind of put some of this stuff together, right? And we talked a little bit about the fact that saturated fats really get a bad rap. This is going to be something that we will take a little bit more of a deep dive on when we talk about questions for take home. Why saturated fats are important. Again, we highlighted in class, your body makes lots of saturated fats. Your body's not going to make something that's bad for you. Same thing with the fact that your body makes cholesterol. So again, we'll take a little bit of a deeper dive on that, talk about why these animal-derived fats are very desirable from sort of a food standpoint, and why the food industry worked to try to convert inexpensive oils into and have the properties of these saturated fats, but how that chemistry really led to the production of trans fats. So we'll talk about that story and sort of the relationship between these different species. We'll spend a little bit of time just kind of talking about the nutritional components of talking about monounsaturated and polyunsaturated fats. Again, bringing in this idea of omega-3 and omega-6 fatty acids and talking about their structures and thus their functions. So when we think about, again, these omega-3 and omega-6 fatty acids, again, anti-inflammatory is what I want you to think about with these omega-3s. So these are a really important sort of nutritional supplement or component for somebody who has autoimmune disease. Omega-6s are important for inducing inflammation. But again, what makes these problematic is they're easily oxidized. And so we don't want to be heating these because that actually increases their pro-inflammatory response. And we talked about the fact that a lot of these come from seed oils, so they're fairly inexpensive, so they're used quite pervasively in processed food products. And statistics indicate that probably about 20% of the total caloric intake from the standard American diet comes from these sort of unhealthy seed oils. Natural sources, again, for these omega-3s are wild salmon. We talked about why farmed salmon, because of the diet they consume, does not tend to be very rich in omega-3s. And again, these animals don't necessarily make the omega-3s. They eat the plants that do make them. So krill is, again, another natural source of omega-3s. So we'll take a little bit of a deeper dive with this story on our take-home question. So some essential omega-3s are ALA, EPA, and DHA. So they just differ by 2 carbons, so ALA being 18 and then 20, C20 for the EPAs, and then DHAs are C22s. And so what's interesting about this story is these larger ones are the bioavailable forms, but this omega-3 tends to be what people consume, is these seed oils. And so we can, but fairly inefficiently, convert these into their larger, more bioavailable precursors. But again, this efficiency is only about 10%. So even if we consume a large number of seed oils that contain some omega-3s, they're actually quite rich in the omega-6s. And so the amount that you'd need to consume to produce these bioavailable omega-3s is kind of not going to get you there. So some numbers that we talked about, ideally you'd like to have about a 1-to-1 ratio between these omega-3s and omega-6s, and that's just generally not realistic with kind of the foods that are available today. So sort of a goal is to have about a 1-to-4 ratio. And there's blood work that you can do that can actually test these ratios. And sadly, there's many individuals who even think they're eating quite healthy who have these ratios of 1 to 20 or more. So very, very heavily promoting these omega-6 fatty acids, which leads to this inflammatory state. We talked a little bit more then about going to these smaller fatty acids. So again, looking at fatty acids between 6 and 12 carbons, we call these medium chain triglycerides. That's what this MCT stands for. What's really important with these from a dietary sense is these are broken down via a different enzymatic pathway than longer fatty acids, and they generate energy very quickly, often by the quick production of these molecules called ketone bodies. So I'll get to those in just a minute. But some important properties of these smaller fatty acids that are found in these medium chain triglycerides are they have significant antiviral and antimicrobial properties, which is, again, really important when we think about the health of your microbiome. So we start moving down into even smaller than 6 carbons. We get to this 4-carbon piece, which is called butyrate. So this is a small chain fatty acid. And so this is a really important food, if you will, for your colonocytes, for the cells that are found in your colon. And small chain fatty acids are actually produced when the prebiotics, the food that you put in for the probiotics, the bacteria in your microbiome, when these bacteria convert this food into small chain fatty acids. It's a really beneficial thing because you're providing fuel for your colonocytes. So I'm going to spend a little bit of time talking about these ketone bodies. We're going to take a much deeper dive next semester, maybe a little bit in a take-home question here. But not structures or names that you need to know, but I want you to be able to look at these structures and understand that they are related. So for example, these two are related by redox chemistry. So again, acetoacetate is the oxidized form, beta-hydroxybutyrate is the reduced form. Acetone is the decarboxylated version of acetoacetate, which is what we call a beta-keto acid. And so this group can readily come off as CO2, and then you're left with acetone. So why you have these different flavors is if you happen to have an excess of ketone bodies, there's a couple ways that you can try to get rid of it. Again, you can decarboxylate it so that this becomes the more volatile acetone that you can exhale. Or again, you can interconvert between these two, which might allow for better excretion. So as we mentioned before, though, these are acidic molecules, so they can, through sort of metabolic acidosis, decrease your blood pH. So somebody who's producing a lot of ketone bodies, and part of your take-home may cover topics related to diabetics and the production of ketone bodies. But this is something that we need to be conscious of so that we're not getting ourselves into a problematic pH state in your blood. So when we take these fatty acids and we hook them on a glycerol backbone. So we never really showed or looked at the structure of glycerol. So glycerol is 3 carbons here, and then these are each OH groups, so a triol. And then again, you're going to have these fatty acids that have a carboxylic acid functional group. Mixing carboxylic acids and alcohols will give you esters. So 3 functional groups I want you to know with regards to fatty acids and triglycerides. So when we link these all together, again, this is what we kind of represent by 3 fingers here, this triglyceride. These triglycerides are found in both exogenous and endogenous sources. So when we're talking about dietary fats, things that we consume in our diet that we obtain externally, we refer to these as exogenous. If we're talking about the fats that you actually have stored in your own body, in your adipose tissue, these are endogenous. So again, this is an internal source of triglycerides. This is how your body stores excess energy as fats. So again, the linkage that we have here is an ester linkage. So if we're talking about how we make that linkage, it's condensation, again, of a carboxylic acid and an alcohol functional group. And so triglyceride synthesis is how we're going to make this linkage. When we talk about breaking this linkage, and you have a couple of reaction mechanisms that are dealing with this, It's a hydrolysis reaction to break this ester linkage, and again, we'll get back our free fatty acids and then our glycerol backbone, so our alcohol there. We talked about in lab the enzyme esterase, which is responsible for cleaving this reaction, or cleaving this linkage. And then we also talked about, and we'll see in a couple of slides here, how we can use this hydrolysis reaction to take what is essentially a fat. We can hydrolyze it to get free fatty acids, and that's how we make soap. Sort of an important theme that we will continue to see here. If you consume something in your diet and you want to uptake it from the gastrointestinal tract, you need to convert it into its monomeric form, because that's the only way you're going to get it through the channels, through the gates, if you will, on those single cells that line your intestinal tract. Now again, if you've got leaky gut issues, if that's a problem, then larger molecules can get through, but again, that is more likely to elicit an immune response. This is a theme of something we've seen before, but we're going to highlight it again here. If you have excess levels of glucose, that's toxic. That's going to promote the production of insulin. That insulin signal is going to cause your liver to take in that glucose. Once those glycogen reserves in your liver have been sort of stockpiled and filled up, any excess glucose needs to be converted into triglycerides. So again, you can convert glucose into fats here, and then those fats will be exported from your liver, stored in your adipose tissue until a time when you might need them. And when you need them, you can then break them down and use them as fuel. So again, the problem is, and we saw this before in our carbohydrate chapter, pervasively high and chronically high insulin levels due to insulin resistance does not allow you to tap into these fuel reserves. Because remember, high insulin levels, they stimulate triglyceride synthesis and storage and inhibit the release of those. So even if you are in a situation where you might need energy, if you have high levels of insulin circulating in your blood, it's going to inhibit this process. So a couple reaction mechanisms I'm going to skip through because we haven't yet had you guys get through to them in the ones that you have that are due daily. So we won't talk about the details with them. So the last couple of slides have to do with sort of thinking about structure-function relationships with these fatty acids, and not only the fatty acids by themselves, but when we have 2 fatty acids on a glycerol backbone, that gives us phospholipids. 3 fatty acids, again on a glycerol backbone, represent triglycerides. So let's start over here with free fatty acids. So again, how we get free fatty acids is we have triglycerides, and we can break that ester linkage. In reaction mechanism, I think it's 18, looking at the base-promoted hydrolysis of triglycerides of that ester linkage, will generate free fatty acids. So if you remember what free fatty acids look like, it's got a large hydrocarbon tail, so very hydrophobic, and then a hydrophilic head group in that carboxylate group. Well, if we remember the hydrophobic effect, right, the tendency of nonpolar substances to minimize their contact with water, we're going to have these guys associate so that their hydrophobic tails will all be next to one another, and they will form the center of a spherical shape called a micelle, where it's surrounded on the outside by these polar head groups. So we have these little spheres that are greasy on the inside, hydrophilic on the outside. So that hydrophilic outside makes these little spheres very water soluble. And so what we can have is then a greasy interior that can pull dirt inside, and because the particles are water soluble, we can wash it away. So this is essentially how soap cleans, right? And I like to think about, well, that's really kind of what the picture of scrubbing bubbles looks like. So we're going to spend a lot of time talking about phospholipids in chapter 10 when we talk about membrane transport. So we'll revisit this then. But again, if you've got phospholipids, you've got 2 hydrocarbon tails here. You still have a hydrophilic head group. We'll spend some time in chapter 10 talking a little bit about how you can decorate this head group with different functionality to change sort of what you have on the surface. But at the end of the day, the shape of this is such that you want to have this bilayer. You want to have sort of a linear array where the hydrocarbon tails are towards the center and the hydrophilic head groups are towards the outside. And this allows us to generate a sidedness to a lipid bilayer. So our lipid bilayers are going to have insides and outsides. The last piece that we're going to talk about is triglycerides. And this structure here is not a triglyceride. It's actually kind of an expanded micelle here. made with phospholipids. So again, if you imagine this guy getting bigger, you wouldn't want water to be in the center. But if you had some greasy stuff that you wanted to get in the center, this could become a larger and larger structure. And so these are vehicles that we're going to use to transport lipids, right? So we're going to have these nonpolar passengers that are going to travel around in this interior. And one of the passengers that likes to be in this interior are triglycerides or triacylglycerols. We're also going to see cholesterol sort of making up the outside layer of this. And that's because it can serve not only as a mechanism for transporting cholesterol, but cholesterol serves as a plasticizer, or something that helps us modulate the fluidity of this structure, such that we're going to have have a transition temperature, a melting temperature that's not a narrow range. We're going to have sort of a larger range there. And we're going to expand on what this purple thing is on the next slide here. But this represents a protein that sort of wraps around this vehicle. So these vehicles are called lipoproteins because those are the two components that make them. And this protein component that we have here really provides directions for where these particles, these vehicles, are going to travel. So again, a lot of this is going to have to do with transport of cholesterol in your body. So again, we highlighted that cholesterol is a really important biomolecule. We hear about cholesterol being good or bad, or the good cholesterol and the bad cholesterol. I want to highlight that cholesterol is an organic molecule. This is the structure of cholesterol. We can see it's a steroid backbone, and that's going to become important because it's actually a precursor for lots of hormones that you need in your body. But it is an organic molecule. What makes it quote unquote good or bad is the vehicle that it's part of, and again, only if it's in high concentrations and in what we call a pro-inflammatory environment is it really considered bad. So let's kind of step back here for a second. So again, it's an important biomolecule. About 30% of your lipid bilayers are actually going to have this cholesterol. And again, that helps modulate the melting temperature of that lipid bilayer so it maintains its fluidity. There are lots of downstream uses of cholesterol. We'll take a deeper dive in some take-home questions, but we'll also spend time talking about this again next semester. But we're going to spend some time talking about statins. So I do want you to know that statins are a class of drugs that inhibit the synthesis of cholesterol. So statins do exactly what they're supposed to. They inhibit your body from producing cholesterol, which will lower your cholesterol levels. However, it also inhibits the production of all of these downstream molecules, and that's wherein it can become problematic. So we'll spend some time in your take-home kind of addressing some aspects of this. So again, that's a problem that I see with the overuse of statins. So again, breaking out into what we typically think of as LDL cholesterol being bad and HDL cholesterol being good. Remember, cholesterol is just an organic molecule. HDL and LDL just represent different vehicles for the same molecule. And we'll see on the next slide that the directions for the where these vehicles travel are what makes them different. So the LDL particles have proteins that direct their travel from the liver to the tissues. So again, these are inbound to your body. And so it's kind of putting cholesterol into your body. If you want to get cholesterol out of your body, HDL is responsible for that because its direction takes cholesterol from the tissues and takes it to the liver where it can be disposed. So again, I want to highlight that cholesterol is really only bad if you have high levels of it and if you're in a pro-inflammatory environment, because this is what leads to things like atherosclerosis and kind of the hardening of the arteries by making plaques. So again, we'll take a little bit of a deeper dive with that with some take-home questions. So no one kind of argues that HDL is sort of a good cholesterol carrying species because of its direction to kind of allow excess cholesterol to be removed from the body if needed. But I do want to highlight this fact that LDL sort of gets a bad rap. And when we think about LDL, importantly, there's at least two main types, or I should say there are two main types, and then there's some subcategories within these two main types. The A type of LDL cholesterol represents these large and fluffy particles, which are actually really important for the healing process. So cholesterol is a really important molecule that helps heal when there's an acute injury or trauma. So again, just to give a little analogy, we didn't talk about this in class, but imagine that there's a fire, there's something terrible going on, and somebody just kind of drives up and they see the fire going on and they see the firemen there. Well, you wouldn't necessarily make the assumption that the firemen caused the fire or they're doing anything bad. They just happen to be there. And if we really think about and know what the firemen's doing, they're actually trying to put out the fire and help with this problem. And so cholesterol has kind of gotten a bad rap because when we have inflammation in our arteries and we have plaques that are formed, people noticed that there was cholesterol there. And so they assumed that it was the cholesterol that was causing the problem. But what we don't really realize, it's the cholesterol that's trying to heal that inflammatory problem and that kind of deposition of cholesterol that's there. So again, this healing, in terms of serving as a fireman, sort of got it a bad rap because it was just identified that it was there when we really didn't know what it was doing. So cholesterol has a lot of important healing properties. Here's where it becomes problematic. There's another type of LDL cholesterol that is what we call small, dense LDL. And the problem with this is it's very prone to oxidation. And lipids that are prone to oxidation increase sort of your inflammatory response to those molecules. And again, if we think about inflammation as being what's problematic, the more inflammation you have, the more problematic it might be if you have elevated cholesterol levels. So the last thing we kind of talked about with cholesterol particles was kind of thinking about the different vehicles that we have. So chylomicrons, we'll take a deeper dive with these when we talk about lipid metabolism next semester, but these are very big vehicles. Okay, they're very, very low density, and that's because they have much more fat compared to protein. And so here we've got these large chylomicron, and again, they've got this A1 protein, and that A1 protein directs them from the intestines to the liver. So these are what our dietary lipids are. If we notice another particle over here, HDL particles, these have the same protein on their surface, so they direct things in the same direction. They take things from the tissue to the liver. When we have LDL and VLDL particles, the main difference here is whether these are full of triglycerides when we have our VLDL particles, so these yellow are triglycerides, or if they've actually delivered them to the tissues, basically the only thing that's left is what we call these cholesterol esters. So this is a very cholesterol-rich particle. But you can see the directions for these are to take them out to the tissues. So in essence, you have a whole bunch of these buses that are full of passengers like cholesterol and triglycerides, and unless they have some place to go and a need at the tissues, they're just going to continue to circulate in your bloodstream. And unless you have, high levels of inflammation around, these are not likely to be problematic. But if you have high levels of inflammation, these can be problematic. So the last little thing that we talked about in this chapter had to do with the fact that, lipids are sort of a catch-all, and I wanted to talk about two fat-soluble vitamins that are really important and have significant implications with regards to cardiovascular health. And so here's vitamin D, specifically vitamin D3, and we're going to talk about this in the context of its importance to calcium homeostasis. We've got vitamin K1 and K2. K1 is the one that's really important for clot formation. We're going to focus, this is K1. We're going to focus on K2 here, and again, all that's different is this R group. So K2 is, again, really important for calcium homeostasis. And if we haven't talked about what the word homeostasis means, that's just kind of maintaining the physiologically correct concentrations. So we've clearly been talking about calcium homeostasis. So again, calcium is, as we know, an inorganic cation. But what's really important about it is it's a very important secondary messenger. So it allows us to kind of transduce a message, so signal transduction. We have something going on the outside of a cell that can trigger the release of calcium either in or out of the cell, and that can serve as a secondary messenger. Muscle contraction is reliant upon calcium signals. Certain enzymes utilize calcium for structure and or catalytic function. But one of the things that you probably already know about calcium is it's important for kind of mineralization of bones and teeth. So I'm going to kind of expand here on why we need to think about vitamin D3 and vitamin K2. So obviously we can't make calcium, we can't make an inorganic mineral in our body. So the only way to get calcium into your body is to consume it in your diet. So whether it's milk products, dairy products, if somebody told you as a woman that you should be taking a Tums every night to get sort of the calcium from the calcium carbonate, we've got this ion in our intestines, and we can increase its uptake by about 25-fold if there is vitamin D3 around. So this helps package and pull in calcium from your intestines. And that's really great because now we've got calcium in our blood that we can use for all of these important functions. However, if we want to have mineralization of our bones and teeth, we have to make sure that we have sufficient vitamin D3 and K2. These two together are required to remove calcium from the blood and then deposit it as minerals in bones and teeth. So if you have either excess calcium and or you are deficient in vitamin D3 or K2, you're going to have this calcium build up in your bloodstream, and that's what we refer to as hypercalcinemia. So hyper meaning high, calcium, and then emia refers to anything that's in your blood. If we talk about urea, that's going to be in your urine. So high calcium blood levels. And what can happen is if calcium levels get too high, your body tries to do something with it to get rid of it. Just like when you have high glucose levels, your body tries to get rid of it in some way by, converting it into fats. In this case, if you've got high levels of calcium, your body will end up depositing it in soft tissue, not in mineralization of these hard surfaces, but it'll throw it into soft tissue as calcium deposits. So again, this can lead to tartar production. So we talked about in class, if you're a medical professional and you have a patient that has significant tartar issues, it might be an indication that they've got problems with hypercalcinemia. They might have a vitamin D3 and or K2 deficiency. And this is sort of the outward signal of what's sort of going on there. Again, deposition of calcium deposits in your vascular system is really problematic. That decreases the elasticity of your vascular system, which can again increase blood pressure. Eventually it can lead to blockage. Deposition in your joints is problematic, and this can also lead to a narrowing of your facial structure. If you're not putting that mineral into the bones and teeth where you need it, as bones grow and as a child grows, if they have sort of this excess calcium that they cannot get into their bones and in their teeth, that can lead to sort of this narrowing of the facial structure. So that was all we really kind of talked about for our chapter on lipids. Again, not a lot that is required for the in-class portion. There's going to be a significant sort of % of your take-home question that's going to deal with applications of this lipid system.